All right, so we just concluded that at a particular synapse, the presynaptic cell is always going to release the exact same neurotransmitters. The postsynaptic cell is going to have the respective receptor, and it's always going to be permeable to the respective ion. Because it's always permeable to the appropriate ion, that effect is either persistently depolarization, causing an excitatory postsynaptic potential, or out of the synapse it may hyperpolarize, which we called on Tuesday an inhibitory postsynaptic potential. Now we're going to go from the more generic terminology and look at some specific neurotransmitters and specific receptors. Instead of just saying neurotransmitter, we'll say acetylcholine, serotonin, dopamine, right, and etc. Table 8.1 summarizes some different neurotransmitters based on their chemical makeup. So chemically, structurally, we can look at some neurotransmitters and call them choline derivatives. So acetylcholine comes from choline. We have some which are called amines, biogenic amines, including catecholamines. When I use the word catecholamine, or anybody uses the word catecholamine, it actually means 3 types of neurotransmitters, or maybe 1 of 3 types. They may be talking about dopamine, or epinephrine, or norepinephrine. Serotonin is also an amine, and histamine, they've heard of that one as well. It's an amine-based neurotransmitter. Some amino acids, which you probably studied for making proteins. Amino acids can serve as messengers in the brain, including glutamate, acetate, glycine, and gamma aminobutyric acid. Other products called purines, which ATP, you've heard of that. We talked about energy transfer. It can also serve as a messenger in the brain. ADP, adenosine diphosphate, can also act as a neurotransmitter, so more than just energy transfer for those 2 molecules. And the adenosine itself, without the phosphate groups, can also have neurotransmitter activity in the brain. Other peptides, this is something getting a little bit bigger than a amine subpeptide. You may have heard of oxytocin. We talked about that this semester. It's a neurotransmitter, neurohormone. You've probably heard of endorphins before. So peptides act as neurotransmitters. Nitric oxide, a gas, can actually have neurotransmitter activity at the nervous system. So this is an idea, it gives you a snapshot that our neurotransmitters are more than epinephrine, dopamine, and serotonin. Okay, so there's various molecules out there that act on the brain, but we're going to just kind of hone in on those really famous ones: dopamine, serotonin, epinephrine, and another one called acetylcholine. The next image that I have for you I found from a previous textbook by Sanders. I like it because it takes these neurotransmitters, which were just shown in that table, gives the structure of them, but also correlates it to the respective receptor families. It gives you a little bit of information about them. So I'm certainly not going to have you go like a biochemistry class and draw these messengers, but I would like for you to understand that acetylcholine combines to how many classes of receptors? 2. And this right here should summarize what we studied in chapter 5. Receptors may be just chemically gated channels, or they may be, we didn't use the word 7 helix, but they may activate a gated ion channel, or a what? Just showed an image about 5 to 10 minutes ago. Clarify just a receptor. Because they are receptors, they can be channels or they may. Yes. A G protein. Exclusively, we said these hydrophilic messengers are either going to open or close an ion channel or activate a G protein. So where it says 7 helix, that's just looking at the structure of it. You can say G protein. So on there, just write G protein. Activate a G protein receptor for the 7 helix. And if you have no idea what I just said, put a big star in there. Go back to chapter 5 and review how hydrophilic messengers activate the target cells. So hydrophilic messengers bind to membrane-bound receptors of the target. Those receptors are probably either channels, translocated channels, or activating a G protein. Okay? If we look at some of these neurotransmitters such as acetylcholine, we can see there's actually how many classes of receptors for acetylcholine? 1, 2, very good. Some acetylcholine receptors are simply just channels, and some are more complex and activating G protein. Whether you're looking at either a channel or a G protein that binds to acetylcholine, you could just, without being specific, you could say an acetylcholine receptor is called a cholinergic receptor. Cholinergic. Receptors bind to what? Cholinergic receptors bind to acetylcholine. Cholinergic is a term to refer to acetylcholine. So cholinergic receptors bind to acetylcholine. Cholinergic doesn't give me any additional detail other than it binds to acetylcholine. So there are two subtypes of cholinergic receptors. One acts as a channel. We are going to call that a nicotinic. Cholinergic nicotinic receptors bind to acetylcholine and cause the postsynaptic cell to become permeable to what? Sodium and potassium. Not the pump, it's a different, this is not the pump, this is a channel. It's a channel, which would be type of passive transport. And so on this one, circle sodium, it's going to let more sodium in than it lets potassium out. So what do you think the target cell would do when a nicotinic cholinergic receptor is open with acetylcholine? The postsynaptic cell would depolarize. Maybe, probably go to a threshold, but we don't know. It would at least first lets more sodium in than potassium out. So if I'm at minus 70 and then I add sodium, my cell will depolarize. So if I open a nicotinic receptor, the cell will depolarize. If it gets the threshold or not, that depends on the amount of acetylcholine. Does that make sense? So the nicotinic receptors are always excitatory at the postsynaptic cell, or I can say they cause the postsynaptic cell to depolarize. You will find these nicotinic receptors in 3 primary areas of the body. One you can handle today, they're at your skeletal muscles. So that means we'll cover this in chapter 12. Your skeletal muscles have nicotinic cholinergic receptors. So that means your muscles can respond to acetylcholine. When acetylcholine is present, your skeletal muscles will depolarize. And it should be enough to let it to get the threshold and generate enough potential. So we can handle that. We know what a muscle is. The other bits may be unfamiliar until we clarify it in chapter 11. You're also going to find nicotinic cholinergic receptors in the middle of your adrenal medulla. We'll cover that in chapter 11. So the adrenal medulla will depolarize in the presence of acetylcholine. And then a third location out in the periphery of the body, where you're going to find the nicotinic cholinergic receptors. This is going to be a new word to you. It won't make sense till chapter 11, but on what's called the post-ganglion cells. Of our, I'm going to abbreviate, autonomic nervous system. So again, chapter 11. So next week, we'll put these nicotinic cholinergic receptors to use. But the postganglion cells will depolarize the presence of acetylcholine because they have nicotinic receptors, right there, which are chemically gated sodium channels. So acetylcholine can activate our nicotinic cholinergic receptors. And we see it not exclusively, but used later in this presentation or class. How to activate our skeletal muscles in chapter 12. How to activate our adrenal medulla, covered in chapter 11. And then the rest of the postganglion cells of the autonomic nervous system also addressed, coming up in chapter 11. We can now also take away that acetylcholine doesn't have to bind to just nicotinic receptors. It can also bind to other receptors called, what we call these? We're going to take a stab at that. Muscarinic. Okay. Muscarinic, that's how I enunciate it. Muscarinic receptors are G protein receptors. And so what that tells us is they aren't as simple. You're like, that was simple? Kinda, because skeletal muscles, the adrenal medulla, and the postganglion cells will always be polarized. When we activate G protein, you may recall covering in chapter 5, this may happen, that may happen, this something else may happen. Who knows? It's pretty darn variable. And then so the rest of the cells in the central nervous system, where you will find muscarinic receptors, they may do what? They may depolarize. Completely other cells with the same muscarinic receptor may hyperpolarize. So it's variable because we're using a G protein. The muscarinic receptors give us more complexity in activating various target cells in presence of the same neurotransmitter, acetylcholine. These, what we just talked about, is summarized. If I don't have the figure number on here, acetylcholine receptors shown in the first left. Which one would this be? Chemically gated channel. This must be a. So it's not a pump. It's a nicotinic receptor. You don't want to use the word pump because what type of transport is going through here? Passive transport. What kind of transport is pump? Active. And so this is an active transport. You know, the pump is pumping. You could draw the sodium potassium pump next door, but when we open up a nicotinic receptor, it's a form of facilitated diffusion. And then so sodium would flow in more, and potassium would flow out. And so the effect of this chemically gated sodium channel is the target cell would depolarize. So it's always excitatory. The image on the right, subset B, is illustrating, still we can see it's a cholinergic receptor because it's binding to acetylcholine, but it's not a channel. We can see what's associated with it. A G protein. Which if you go on and open some ion channel, notice it's non-specific. It could be sodium in some tissues and potassium in the other, or it may activate a more complex second messenger system. That's the diversity of using a G protein. Those you would collectively call muscarinic receptors. What questions you have about using the word acetylcholine, cholinergic, or the difference between a chemically gated channel here, like our nicotinic receptors, or the application of a G protein like we see here with the muscarinic receptors. This is just an example of what we said could happen in Chapter 5. Since we're just getting specific chemicals here. Another note that I have here is, Matt, take it away. Acetylcholine is used certainly in the peripheral nervous system. We gave 3 tissue examples over there, but it's also important in the central nervous system as well. So it's throughout the body you would find the acetylcholine used. Because it's used in the peripheral nervous system, that means we'll see it, and that's why we're taking so much time to talk about it. We're going to see it again later in the rest of the body. Another neurotransmitter we can talk about is called dopamine. Dopamine is a neurotransmitter that falls into our category of catecholamines, which we listed a moment ago. How many classes of receptors could dopamine bind? One. And its receptor acts as a G protein. So that tells us, can we keep it simple? No. May excite some tissues, may inhibit others. But its name's not too tricky. It's called a dopamine receptor. Other neurotransmitters, another catecholamine is called norepinephrine. We gave an example of this previously when we talked about how G proteins can bring about various responses. So we see this table backs it up. Norepinephrine binds to G protein receptors. Their receptors are called adrenergic. That one kind of sounds like a bit of a tongue twister. Adrenergic receptors bind to norepinephrine. And they do associate with the G protein. They bind to both. The name may look unrelated, adrenergic, norepinephrine, but actually, if I give you the name of this neurotransmitter when it's in the blood, what do you think I call it? Adrenaline. Adrenaline, and there's a variant of it called noradrenaline. So if you called it adrenaline, it makes sense that it binds to an adrenergic. You see that prefix commonality? Adrenergic receptors bind to adrenaline. Adrenergic receptors bind to noradrenaline, norepinephrine, epinephrine. They're all, the difference is it in the blood or in the synapse. You would call it adrenaline or norepinephrine. So adrenergic receptors do activate G proteins, which may cause some cells to excite, may cause some cells to inhibit. We gave an example of epinephrine at the liver tissue, where we saw glycogenolysis happening, gluconeogenesis. In fat tissue, our adrenergic receptors cause lipolysis. Norepinephrine and epinephrine at some smooth muscle binding to an adrenergic receptor causes some smooth muscle to relax in parts of the body, but do what in other parts? Contract. Because we're using a G protein, which gives us some variability in responses. Another neurotransmitter that you can talk about is called serotonin. It's an amine-based neurotransmitter, and it can bind to how many classes of receptors? Two. Some serotonin receptors are what? Channels. Okay, so they're gonna. It says excitatory, so they would cause depolarization. And some serotonin receptors, called serotonin receptors, also activate G protein, because they may cause some cells to
depolarize or hyperpolarization.
When activated. What questions do you have at this point before we kind of go back and give some additional detail on these monoamines? To add to your enthusiasm, which I can see here, the adrenergic receptors, did I just say adrenergic? That's a family of receptors, and so if we use the family, that tells us what? There's a family. There's more than one type. Okay? So if you think about your family, there's more than just, hopefully, may not be, but more than just you, right? right? You or maybe a parent or elder and a sibling or something like that, right? So same thing with the adrenergic receptors. There's the alpha family, the beta family, and the alpha family has subunits. The betas, there's alpha 1, alpha 2, beta 1, beta 2, beta 3. They would all be alpha adrenergic, beta adrenergic, whatever, right? The specific is that it continues or allows for these various responses in various tissues. If we follow along with our guided notes, we mentioned earlier that some neurotransmitters may be categorized as monoamines, including serotonin. It comes from tryptophan, and other catecholamines. We said the word catecholamine could collectively include epinephrine, norepinephrine, and dopamine. In this class, I may just say epinephrine, but I could say epinephrine and norepinephrine.
So they have the same receptors?
They have the same family of receptors. They would use alpha adrenergic, beta adrenergic receptors. They're very structurally similar. Norepinephrine is more commonly used. Numerically, I don't know what the percentage is, but numerically it's more. But I tend to just say epinephrine. It's easier for me to say. And so when I say epinephrine, you could also say, and she also means norepinephrine. They say each bind to adrenergic receptors. Dopamine is also a catecholamine, and it binds to not adrenergic, dopamine receptors. Norepinephrine and epinephrine exclusively would bind to the adrenergic receptors. Dopamine binds to dopamine receptors. Serotonin binds to serotonin receptors. Acetylcholine binds to
Nicotinic.
you can either say nicotinic, you can say muscarinic, or you could just say cholinergic. Acetylcholine binds to cholinergic receptors. The two families below would be nicotinic and muscarinic. These monoamine neurotransmitters are probably the neurotransmitters which you knew about before coming to class. You've probably heard of dopamine. You've probably heard of serotonin. And if you've not heard of epinephrine, you've heard of adrenaline, right? You've heard of these things. These categorically are structurally called monoamines, so these small amine-based molecules. And in the brain, when they're released, you recall, once a messenger is released, does its job, it needs to get cleared out so it doesn't falsely stimulate the target tissue. Okay? We don't want some neurotransmitter. Let's think about, let's give a non-brain example, brain target example. Oxytocin in the female uterus, right? You wouldn't want a woman having a baby to start the contractions and they keep going and the baby comes, but then after the baby and the placenta are delivered, you don't want that messenger binding to those muscles and then sustaining contraction indefinitely, right? You wouldn't also want the same thing in the brain to have the brain cell receive a chemical messenger and just keep responding. Because it keeps responding, it's not responsive to a subsequent stimulus. And so those messengers need to be taken care of. And one way that these monoamine messengers can be taken care of or cleared is by using an enzyme that's called monoamine oxidase. We work through it backwards. It's an enzyme that oxidizes monoamine. Fair enough? You've probably never heard of that, but you may have heard of MAOI. Have you heard of those before? You watch TV? So I see the sad person with the balloon. Right. We talked about MAOIs for what? Antidepressants. You see those kind of spewed all over the media. What that means is these are inhibitors to an enzyme that does what? The enzyme doesn't make you feel sad. What does the enzyme do?
Getting broken down, so neurotransmitters.
Right. And so the inhibitor inhibits the enzyme from breaking down the neurotransmitter. And if we look at some serotonin and dopamine, these do have a lot to do with mood and behavior and emotional responses. And in so often cases, if these levels are low in one's body, and which they're already low, and you have an enzyme to break them down, they continue to be low. So a doctor could prescribe you an inhibitor to that enzyme. Your low levels of neurotransmitter, what's the effect?
Stop breaking them down.
They stop breaking them down. They stay in the. They don't go quite into the blood. They're just at a synapse. They're neurotransmitters, so they stay in the synapse. They can continue to bind to receptors and continue to have the appropriate response on the nerves in your body, in your brain.
Wouldn't that be bad over a long period of time, though?
Not necessarily. If your levels of serotonin and dopamine are below set point, this is a kind of a quick way to bring those levels, if you will, back to set point, at least the activity, the stimulation on the brain. It would be bad if someone's taking these and it's not prescribed and they don't need it. Yeah, that would be bad. But replacing, or I guess giving this inhibitor to those enzymes, if those neurotransmitters low, no, that's okay. And not just antidepressants, that's what we see in marketed. You've got other brain diseases like the headaches, the migraine headaches, for example. You may see these prescribed to kind of keep the neurotransmitter balance back at set point. We already kind of addressed norepinephrine. I said also, I can say epinephrine. We identified that there are two classes of adrenergic receptors. They're called alpha and beta adrenergic receptors. And here, never say never or always. So we can say, well, generally, my abbreviation, norepinephrine, when binding to an alpha adrenergic receptor, is generally stimulatory. Generally, not always. And while epinephrine and norepinephrine combine to beta adrenergic receptors, the effect is usually inhibitory. So usually. A very important notable exception is probably what you knew coming into this class. You didn't know that you knew it was called an adrenergic receptor. You work in a medical facility, you ever talk about beta blockers? You've been in the hospital, heard about them, beta blockers. It's a lot easier to say than beta adrenergic receptor blockers, right? It's easier to say beta blockers. So a doctor may prescribe a beta blocker, a beta adrenergic receptor blocker. When might this be prescribed? What are you blocking? When a patient's blood pressure is too high, or maybe they're in the middle of what? It's like a cardiac failure, right? Beta adrenergic receptors on the heart do what to the heart rate? Increase it. And the force of contraction is also increased. That makes sense, right? When you're exercising, epinephrine is going through your blood, your heart rate's going to go up, your blood pressure is going to go up, which is good, you're exercising. But if you're taking a nap, blood pressure goes up, heart rate goes up, you're probably in the middle of a heart attack. And so the doctor could give you a beta blocker. So even though those levels are high, you block the receptor, and the heart should hopefully do what? Relax a little bit, bring that blood pressure down, bring that rate of contraction down, hopefully back at least closer to set point, if not within the set point rate. So the exception is beta adrenergic receptors at the heart are actually what? Stimulatory. All right, well, that looks like that concludes good old chapter 8 for us. All right, we're going to enter into or discuss chapter 9, which is dealing exclusively with the central nervous system. The bulk share of the material covered here in chapter 9, we covered for nearly 3 hours last week when we did our brain dissection, brain anatomy, spinal cord anatomy, and physiology. So a lot of it is going to be repetitive, and the purpose of having the lab in advance so we can bring up questions, specific questions, instead of being overwhelmed. If we look at chapter 9, we're going to talk about the structure and the function of the blood barrier. This is new to us. We didn't talk about this in lab, and as well as the nutrient sources for the central nervous system. Second thing we're going to talk about is the overall anatomy of the brain and the spinal cord, again, repetition from lab. We're going to review the pathway of a reflex arc, which was kind of warmed up to in lab last week, presented in lab this week. We're going to carry out some actual reflex arcs. And then reviewing from lab last week, we're going to talk about the parts of the brain involved with voluntary movement, somatosensory processing, visual processing, and auditory processing. So let's just crash course. Voluntary movement is in what part of the brain? We're in the central nervous system. What part of the central nervous system? The frontal lobe. It's going to talk of things like the cerebellum, coordinating movement. Somatosensory, parietal lobe, visual, occipital lobe, auditory, temporal lobe. So we're just going to review those things. We're also going to repeat very quickly the relationship of the Wernicke's and the Broca's area for language formation. And then we'll conclude Chapter 9 information, if not today, on Tuesday, about how to differentiate between what's considered a short-term versus long-term memory, and then see what a person would have to do to make that conversion between a short-term and long-term memory. There's some interesting information about the brain presented by your textbook, is that the brain and the spinal cord, is that you will find each gray and white matter in both the brain and the spinal cord. So that tells us that there's myelinated cells and there's non-myelinated cells in the brain. The brain itself has guesstimated about 100 billion nerve cells. You begin to think each nerve cell can go out and talk to multiple postsynaptic cells, and each postsynaptic cell can be talked to by multiple presynaptic cells. The amount of intracellular communication is almost infinite. How many connections can be made within your brain? The average brain ranges from 3 to 4 pounds. Generally, anatomically smaller people tend to be females, have this lower end, and then larger people would have larger brains, just a little bit larger. If you think about that mass, 3 to 4 pounds, and the distribution of blood, the brain itself receives about 15 to 20% of your total blood flow. So let's go to the easy math. 20% is a fifth of the blood flow. If your brain weighs 4 pounds, is that 1/5 of your mass? 4 times 5 is, did you weigh 20 pounds?
Maybe on the move.
The day I was there, yeah. So, what can we take away? Your brain's 3 to 4 pounds. It receives 15 to 20% of the blood flow. What can we extract from that?
A lot of blood flow goes to the brain.
Yes. So a lot of blood flow goes to the brain. What else can we deduce from there? It's really, really, really important. The brain, the central nervous system needs a constant supply of blood to deliver what? Oxygen and glucose. Predominantly, messengers will be delivered, but that's not the critical part. Before they can receive anything, they've got to leave cells. These hundreds, billion cells have to live. And so they need oxygen and glucose to carry out aerobic respiration.
How is the brain affected by a high protein diet? We're not getting a ton of glucose. Is that,
Yeah, so if you have a diet that's excessive in one nutrient source and lacking in other nutrient source, it clearly can affect all of your tissues, including the brain. A high protein diet, you wouldn't want to have, I don't know of anybody that would have exclusively like an animal protein diet. And so someone is going to get other nutrients, glucose, fats, or whatever in their diet, right? But a diet high in proteins, when we break down the proteins themselves, they can be broken down to things that are potentially damaging to, say, the kidneys, for example, and make the environment kind of toxic. And so a high protein diet may not necessarily starve your body of glucose, right, directly in that fashion, but the byproducts of the protein metabolism could be catastrophic. And so it's not the direct you're not gonna not have glucose in your body if you aren't in one of these concentration camps being starved to death or something like that. You're eating, you're probably taking in nutrients, for example. But on that note, you wouldn't want to have a diet unbalanced. If you want to go on the Atkins diet or whatever, however they want to call it now, the paleo diet, whatever, however you want to call it, or it's high in something, you want to teeter-totter in balancing your protein, whether it be from animal or plants with other fruits and vegetables or something like that. So, balance is good. All right. So figure 9.2 helps us visualize the protection of the central nervous system. I'm just going to show it for your appreciation. When you get into anatomy, you'll go through and look at the fine anatomy of these different structures. But because our brain is important, because it receives this disproportionate amount of blood flow, it's important so we can kind of protect it as well. So not only give it nutrients all the time, but physically protect it. We can physically protect it by using the skull, so the bone around it, and in the spinal cord itself, protected by the vertebrae of your spine. And then also these various connective tissues called matter, the dura mater, the arachnoid matter, and the pia mater. Give physical protection. Additionally, you would have fluid around and within the central nervous system you would call the cerebral spinal fluid. So it gives a cushion barrier as well. So these organs are very well protected, so that we don't randomly damage them. Sitting or sleeping or whatever the case may be. With that, if we continue off the discussion of the blood flow to the brain, we can look at the anatomy of the vessels of the brain. Fully aware that we have not addressed blood vessel anatomy. We won't get there until chapter 14, so this will help us when we get there. But your capillaries, we've kind of introduced this a couple of times. Certainly when we introduce how fluid is exchanged between the plasma and the interstitial fluid, right? We said you kind of move the fluid from one location to another. We're looking exclusively in the central nervous system. When fluid is pushed from a capillary lumen out into the interstitial fluid, you could call it interstitial fluid, but it'd be more specific to call the cerebral fluid, because it's now, it's specifically in the brain, or the spinal fluid, we call it the spinal fluid. And so the way that this fluid is formed is due to the mechanics or the makeup of our capillary cells. And so this is a given capillary. You may recall in chapter 7 we introduced that in the brain we have astrocytes that branch and touch our capillaries and would cling to a nerve cell. So the nerve cells are adjacent or proximal to a capillary. So that our cerebral fluid can serve it back and forth. And so on the top is a long look at a capillary, and the image highlighted in blue is that capillary cut in half and then turned on its end. So we're looking at a cross section of this capillary. Those capillary cells are called endothelial cells. And so in this image here we can see 1, 2, 3, 4 endothelial cells represented. Capillaries of the central nervous system are unique in that there's a high proportion of tight junctions, which we saw covered in chapter, was that 2? We talked about the different membrane junctions. What is the purpose of a tight junction? To prevent leakage, right, between cells. Okay, so these cells are adhered together tightly so that there's not random diffusion between those cells. That's what's going to be unique about the capillaries in the central nervous system, is this high proportion of tight junctions. And so if we want to push the contents of the blood out to form our cerebral spinal fluid, which I just have abbreviated here, CSF. When we get into chapter 14, we'll see pressure. Fluid pressure pushes this stuff out. And so when it originates in the lumen, we would call the fluid what? Fluid in your capillaries. Fluid in your capillaries is called plasma, blood plasma. We push it out force it out, and then in that case, you would, once it's pushed out, you can call it the cerebral spinal fluid. Linking back to what we studied in chapter 4, though, not everything is going to be able to be forced from the plasma to the cerebral fluid, because we've got to look at the size of the molecules and the what? Size in the shape, do it, solubility. So we've got to look at the solubility of a particle, if it's going to be able to diffuse across the membrane into the cerebral fluid. So we review some different types of particles. What about lipophilic substances? Could lipophilic substances transition from the blood plasma and be part of the cerebral spinal fluid? Lipo, fat, loving substances. Lipophilic substances like estrogen, oxygen, carbon dioxide. We want those things. They are able to, right, simply diffuse. Lipid soluble, if you recall, like say our steroid hormones, remember we can't keep them in a vesicle, right? We can't trap them, and so they can simply just diffuse. Lipophilic messengers, molecules like gases, etc., they can be incorporated in the central cerebral fluid, They just simply diffuse out because they're lipid soluble. And the endothelial cells have lipid membranes about them. So chapter 4 and 5, we covered that. So lipid soluble substances do what? Simply diffuse, okay, into the cerebral spinal fluid. What about hydrophilic solutes? No, unless there is a channel or a transporter. So a lot of larger solutes, like glucose, we normally wouldn't be able to simply diffuse, but we could transport it across into the cerebral spinal fluid. So it can simply diffuse things like oxygen into the spinal fluid. We can transport more hydrophilic molecules like glucose out in the spinal fluid. We know that our brain needs oxygen and glucose. What about other hydrophilic substances that may not have a transporter? You could endocytose and exocytose. That is not very common though in the brain. It's not very common, so then our answer would be things like proteins. Will we see them forming in the cerebral spinal fluid? You would not find them in the cerebral spinal fluid. They would stay in the blood because they are too big, and they don't have a transporter. So they stay. Those larger hydrophilic substances, like proteins, they just stay. So I have a big blue protein. It's like rejected. Boom. You're too big. Keep on going through. Does that make sense? Something else that you could draw here that is too big to be filtered out into the cerebral spinal fluid are your big red blood cells. Your red blood cells are almost as big as the entire diameter of the capillary itself, which is good. We don't want red blood cells pooling on our brain. What would that mean? Hematoma. It's hematoma. You have blood on the brain, which is bad, right? So we want to keep the blood cells, red blood cells, in the capillary. So they are they're even larger than this protein. They take up the whole thing. And they're going to stay circulating through the blood. Does that mean they're white blood cells? White blood cells are unique in that they can, remember we talked about, we talked a little bit about phagocytosis in the way that they work. They can also what's called extravasate. That means get out of the vessel. And so they aren't filtered. They would specifically walk, if you will, through. But We don't really need, I don't know how common it is in the brain, because we already have phagocytic cells in the brain and microglia. And so those immune cells are pretty much in place. So I'm gonna guess it's pretty minimal for white blood cells actually extravasating in the central nervous system. Yeah. I'm saying it could happen in any capillary, whether it happens, but it may not even happen at all. I'm going to refresh my memory on that. It may not happen at all in the central nervous system because we do have a microglia in the spinal cord. I'm gonna have to look that up. If the white blood cells get extravasate into the brain, I can't recall right now. But they're different. Even though they're big, they can voluntarily move through different tissues. If we contrast a capillary to the rest of the body, what is different here? So we're looking at some capillary, we can still see our endothelial cells. What looks different here?
No astrocytes.
There's no astrocytes, so we're not in the brain. What else is absent? Well, there are endothelial cells. They're still there.
No tight junction.
No tight junctions. Notice how the cells, the endothelial cells, are arranged with one another? So that we have what? Well, they're called pores. Yesterday, someone used the word gap. I would not use the word gap, so you don't confuse it with a gap junction. These are not gap junctions. These are just where the two membranes aren't held together. Okay, so the membranes are just not touching one another. In a gap junction, if you recall, that's proteins that form a channel. This is not a protein channel. This is just where two membranes aren't stuck together. And so it's almost like maybe think about a sifter, like a flour sifter or something like that. How the wires, they're connected at some points, but there's a gap or a cord, we use our word cord between them. The silver things would be like the cells. So here, some portions, but not all. So there's a cord. So the rest of our capillaries are very porous. And so what would be the difference here? More things are likely to diffuse. What kind of things are more likely to diffuse? Things that are a little bit larger, and or hydrophilic. Assume your hydrophilic substances can just diffuse through the pore. And so we go through and use the same kind of animation as a previous capillary. Out on the rest of the body, you wouldn't call it cerebral spinal fluid, you would call it interstitial fluid, hence the abbreviation here. So the same mechanics we'll learn in chapter 14. Pressure pushes our water and other solutes from the blood plasma to form the interstitial fluid. Same concepts here. Lipophilic particles could simply diffuse across the capillary cells, or they could just simply diffuse through the little pores. And they're even easier for them to get in and out. Hydrophilic substances could still be transported if there's a transporter. And if you're like way down here on the lower right hand side, that's representing that transcytosis we covered in chapter 4. And so you'll see that endocytosis, trafficking, and exocytosis. So you're going to see some proteins or larger hydrophilic particles transcytosed from the plasma across the capillary cell out into the what? Interstitial fluid. You can still see. What?
I was going to say transcytosis just means that both times can happen.
It's using both. Taking a particle, first endocytosing it, then trafficking it, then exocytosing it. So it's just a transport across the cell, this tiny little cell. What about, so we said, even though that's not proportionate, that's just a little peptide. If it's able to fit through the peptide, through the little pore, it can be forced out into the interstitial fluid. What about really large proteins like albumin? What about really big proteins like albumin? Can they be collected in your interstitial fluid? No, because they are too big to fit through the pore, and so they're still rejected. Boom. You stay in the blood. What else stays in the blood? The red blood cells will stay in there as well. Alright, so the capillary, the point of that is that the capillaries are much more restrictive in the central nervous system than they are in the rest of the body. Because the brain and the spinal cord are super, what? Super important, okay? And we don't want just random particles that you may be exposed to or putting in your body, okay? You might put in your body, getting into your brain. Getting into it. If you turn to table 9.1, it gives you a quantification comparing the blood plasma in the second column to the cerebral spinal fluid in the right column. If you look at the glucose in the blood plasma, the range we've seen before, if you're diabetic, you know that range, right? So 100 is the upper end of homeostasis. And the blood plasma, but if you look at in the cerebral spinal fluid, the blood glucose level is a little bit lower in the cerebral spinal fluid. Again, reiterating the role or the importance of constant blood flow to the brain. Our protein levels in our cerebral spinal fluid, what in comparison to the blood plasma? Minute, right? 15 milligrams per deciliter to 45 compared to 7,000, reiterating the restrictiveness of the blood-brain barrier in the central nervous system. Sodium, not too different. Potassium, a little bit reduced, but that should be correlated to what we studied about the astrocytes in chapter 7, where we said they pick up extra potassium, keeping it a little bit low so that the brain cell doesn't get accidentally activated, messing up our membrane potential of our nerve cells. Chloride ion looks a little bit higher, and calcium a little bit lower, but not too far off compared to the blood plasma, and certainly not to the degree of discrepancy like your proteins in your blood sugar or your glucose. And so glucose, if we look at what the brain uses for energy, glucose is the preferred, under normal conditions, the only nutrient source for your brain. And so it requires aerobic glycolysis. Time back to what we said a few minutes ago. The brain needs a constant supply of oxygen and glucose so it can produce what? So it can make ATP. So it can do work like pumping the sodium potassium pump, like making neurotransmitter, like creating calcium gradient, and etc. The brain, the nerve cells themselves lack the ability to store any glycogen. They don't have any reserves. And so if your heart shuts down and stops sending blood to your heart, or to your brain, it's going to run out of glucose. It's not going to have anything. It doesn't have a backup plan. It's like give me blood all the time. And so it's been shown that some of our glial cells do have the ability to store a little bit of glycogen, but it's not going to be enough to get you through a heart attack if nobody's there to help you. You have to keep your heart pumping. Fatty acids aren't used. So we need constant blood supply to deliver a constant source of glucose to keep our brain alive. Does anybody know how long you can go without oxygen to the brain before you? It's like 4 minutes, 5 minutes. Yeah, if you see someone who's been out for an hour, they're
Dead.
They're already off. Yeah. Did you say diseased?
No, deceased.
Oh, deceased. They are deceased. It's too late. 4 minutes. And so, if you see someone going to cardiac failure, first thing you can do is do what? Even before then, if you're trained, you might want to do some CPR. But you or somebody else needs to do what? Call 911. Okay, and then start giving chest compressions. And so, if you've got a pair of people there, you need to get that blood flow. You don't have to do the rescue breaths, but compressions will keep that blood, hopefully enough to keep the brain alive. So medical intervention is available. We can refer to figure 9.6, so looking at to review the spinal cord anatomy, which again we introduced in last week. We'll talk some about this week because it's hard to talk about the central nervous system and not the peripheral nervous system. Hard to talk about the spinal cord and not the spinal nerves. I try to mention to most, if not all labs, and that the spinal cord into adulthood terminates about mid-back. And then the lower portion of the back is not the spinal cord itself, but the various spinal nerves extending down. And the spinal cord is an extension of the brain, and specifically the brain stem. And then peripherally, we would have our spinal nerves branching off of it. When you go to anatomy, you'll probably go through and just locate the different nerves. They're named based on the regions in which they're found, so cervical region, thoracic, lumbar, sacral, and coccygeal. That is important, but we're going to look inside the spinal cord and look at a cross section. And last week we became familiar with some basic orientation. We used the term dorsal and ventral to talk about the front and the back of the spinal cord. On either side, not shown here, would be the spinal nerves, the afferent and the efferent nerves. I tried to briefly mention this term called ascending and descending. What does that term mean? Moving up, ascending, and moving down, descending. And so in this image, it's just taking a cross section, a tiny little section of the spinal cord, cutting it and looking at it. When you do that, you have cut a bunch of nerves that would be either ascending or descending. And so the nerve axons have been cut off. And so think if you were to have like a string cheese, right? You have it this way and you cut a little bit, and you turn it on its side, you would have little portions of the little strings, right? Not strings of cheese in your spinal cord, but portions of nerve axons would be cut. And if we were to go through and pick apart the little sections, some of the nerves would have been conveying up, so that means their cell bodies, or I guess let's say their axon terminals are closer to the brain. And some would be descending, where the axon terminals are farther away from the brain. Is that okay with the ascending and descending terminology? We'll also study that some nerves cross over, they transverse, so that we can have not just two people in one body, right? So you have your left and right side can integrate together. But what I'm going to do is go ahead and conclude our discussion here. If you haven't already done so, you'll need to read chapter 9. You can use your guided notes as a template. But we're going to go ahead and get in chapter 10 as well on Monday, the sensory nervous system. So ideally, 9 and 10 will be ready before we get to class on Monday.
Paper.